Good afternoon and welcome to the Utopia Health webinar from Lab to Clinic. Today we will discover how to transform innovative research into clinical viable diagnostic products. And this session will guide you through the critical stages from early concepts to regulatory submission, covering strategies for overcoming regulatory challenges and compiling a robust technical file. With us today is Dr. Matt Pierce. He is a senior executive and founder of MedTech Market with over 30 years of experience leading innovation, commercialization, and growth across diagnostic, bringing technologies from concept to market and securing investments, launching spin-offs, and collaborating with top universities. A short information about housekeeping rules, which are quite obvious. We kindly ask you to keep your microphones muted during the presentation, and we will take your questions after the presentation. Either you write them in the chat or in the end, and I will read them afterwards, or you can post them, ask them yourself afterwards after the presentation. So that is all about the introduction. Let's begin. And Matt, welcome, and the floor is yours.
Thank you, Simona. So good afternoon, everybody. So as Simona just said, I'm going to cover the basic elements today of what you need to do to take a diagnostic technology, all the way through to market. But just before I start, just as Simona mentioned, I've got a number of years experience in the IVD industry, having worked for Amersham, Unilever, Provalis, general manager at Bio-Rad, before I set up MedTech to Market. And MedTech, just a bit about MedTech to Market, we're an end-to-end outsourcing and translational partner for medical devices and diagnostic technologies for a global market. We're based in the UK, and we have an office under MedTech to Market Europe Limited in the Republic of Ireland. The UK head office is where all of our development labs are and our manufacturing. And then in Ireland, we have our Irish business is basically it acts as an EC representative to allow us to do medical device regulation and in vitro diagnostics regulation submissions into Europe. Okay, so as an overview of the presentation today, I'm going to go through the basics of the regulatory situation and framework for diagnostics, and then I'm going to walk you through the development process and activities and everything that's needed. Well, not everything, as much as I can fit into an hour, of what's needed to take you all the way through to regulatory submission. But also it's more about the approach that you need to take. And then also at the end, I'll talk a bit more about some of the commercial considerations. There's just a cautionary note. A lot of these slides have got quite a lot of content. And the idea is so that this webinar is being recorded, so you can go back and have a look at some of the detail if you need to do it at a later date. Okay, so one of the first questions, and we see this a lot, is fundamentally, is the technology that you're developing a medical device? And so in order to do that, you obviously need to decide whether it will have a medical intended purpose, and then whether it's medical device or an in vitro diagnostic. So I've highlighted here in black and in blue excerpts from the MDR, the medical device regulation, and the IVDR, which are the European device regulations. And I've highlighted in bold anything that effectively points you in the direction of whether your technology is classed as medical. So effectively, if your device is designed to diagnose, prevent, monitor, predict, offer a prognosis, affect the treatment of a disease, and or monitoring of all of that, then it is a medical device. And the distinction between in Europe, the distinction between medical device and in vitro diagnostic is really the item identified in blue, which is is this information obtained by measuring an in vitro examination of specimens derived from the human body. So a lot of medical software, software where you're diagnosing, preventing, monitoring, that software will fall under the medical device regulation, the MDR, because it's not doing an in vitro examination of tissue samples or blood tissue or whatever. So that's the first question you need to ask, and we do get asked this quite a lot. But I'm focusing today on the diagnostic spectrum and the diagnostics. And as I just mentioned to you, the diagnostics can fall under both in vitro diagnostics and the medical device regulation. And I've sort of shown you some sort of kinds of examples. So medical devices tend to be equipment that monitors externally the patient, or are accessories to a diagnostic test, like blood collection tubes or blood spots or medical software to help diagnose. So all of these items on the left are used to diagnose a patient's medical condition, to diagnose, prevent, monitor, etc. But obviously on the right hand side, it's more the traditional in vitro diagnostic, where it's measuring patient samples. So that could be anything from simple dipsticks all the way through to large, fully automated lab analyzer systems. So that's the kind of spectrum that we're operating in. And today I'm going to cover pretty much most of these things, but more of a focus on in vitro diagnostics. But the process is virtually exactly the same. So, and when we're looking at diagnostic components, typically these involve chemistry. So a lot of the time for assays, it might be PCR reagents, immunochemistry, lateral flow strips, electrochemistry, etc. And that's how the biomarkers or the patient sample is being measured. But there are also software that can either be embedded or could be an application based. Electronics and hardware, so that might be electronic instrumentation. There's calibrators, so often you have reference samples that need to be provided as a manufacturer to show that your system is working correctly. Other types of calibrators as well, and then things like collection devices and connectivity options for allowing the patient data to be connected, whether it's through smartphones or whether it's through a hospital network. And then also some additional scanners and accessories like printers, scanners, barcode readers, that type of thing, which are also rolled into the overall system and will need to feature as part of the regulatory submission. Now, when you're developing all of these items, I use the analogy of the development process is like making a cake. The process of making the cake is pretty constant. So you need to do all of the stages of the development process need to be done in the correct order. Otherwise, you won't get a cake. You'll get an omelette or whatever. But the ingredients that go into that cake do change. So they are specific to a particular project. So when you're looking at a development process, you need to consider the process and the ingredients. And so I'm going to take you through the process first, and then I'll specifically look at the ingredients. Obviously, it depends on the project, of what the ingredients you use, but I'll give you examples of how you work that out. The process itself should be pretty much the same, whatever the product. Okay, so if I look at the process now, typically when we're working with a lot of academic and university or clinical groups, most of the research focuses on the two red areas that I've got here. So one of them is on the research program, identifying the technologies, discovering the biomarkers, etc. And The other main focus is on clinically validating those samples, those markers, to demonstrate that the technology works. And then obviously with the aim of publishing. When you're looking at this as a product developer and as a commercializer, there are a lot of other steps that need to be done in between, and the clinical validation, as I'll come to later, is a slightly different thing. And these things need to be done in the correct order. So like I say, it's like baking a cake. You need to follow these processes in order. There's a system called the technology readiness level, so TRLs. And so I've aligned these stages alongside the kind of the tradition, the TRL model. We call them milestones, but basically these are stages that you need to do. Ultimately, at the end of this project, at the end of the development project, when you're looking to submit, the whole process is designed to prove that the product is safe and effective, not the technology. So the two red items demonstrate the technology works. The whole of this project demonstrates that the product works. So it's not just the technology. This is as the customer or the user will find this product, it needs to be demonstrated to be safe and effective. Okay, I'm just going to quickly look at the process. And again, the regulatory oversight, I'm going to talk a bit about regulatory. I'm not going to go into massive detail on the regulatory affairs because I've only got an hour. But basically, each country and each region has its own competent authorities. So each country has, particularly within Europe, for example, have their own agencies through which you will do the technical submission and the technical registration. So, and then obviously the FDA is another example of a competent authority. So in the US and CE marking, pretty much for most IVDs, and I'll give you a bit more detail on this in a minute, but most IVDs require a full technical file submission. So in the US, that's what they call a 510K, where you're actually submitting the technical documentation to show that your product is substantially equivalent to an existing competitor or an existing product. Or if there is no existing product, then it's a de novo process. But if it's a high risk item, then it's called a PMA, a pre-market authorization. And so those are the three methods of going in through to the FDA. De novo and PMA are very similar in terms of process, very high levels of scrutiny with the FDA. The 510K, you are really setting your technical file up to compare it against maybe another alternative system. For Europe, under the IVDR, all technical files are reviewed by notified bodies. And these are companies who act on behalf of the competent authority to review the technical files. And so these are companies like UL, DECRA, TUV, etc. And they will review the technical file to make sure it's full and complete. And I'll be talking a bit more about that later. In the UK, the UK currently has self-declaring for most IVDs. So 80% of IVDs in the UK now are self-declaring still, which is very similar to the old system in Europe. This probably will change over time, but at the moment, and as of this year, that is the latest position. So you often hear about risk classifications for products. So I've put in here medical devices and IVD classifications. Obviously, with this particular audience, the majority of people are interested in Europe and the CE marking standards. And as you can see, medical devices and IVD have different classifications. So if I'm really focusing on IVD, the vast majority of products for European submission fall under class B. So they have a moderate level of risk, and they will be class B. And there are, depending on the level of risk, certain cancer diagnostics, certain blood typing diagnostics where things could go wrong, horribly wrong if they get it wrong. But also some self-testing is a slightly different system. But self-testing can often be higher risk of misdiagnosis, then they will go to C or D. But the majority of products are class B in under IVDR, and that's about 80 to 90% of all diagnostic tests will fall under that classification. In the FDA, they're equivalent to a class 2, and in the UK, the difference is in the UK, most of them fall under the general classification ruling, which is self-declaring. Okay. Right, so let's get into what is needed in the process. So the essential requirements of what's needed. So anybody, any company who is submitting is wishing to put a product on the market in Europe, or classed as a manufacturer, they will need to be certified. They need uh, under the IVDR, they need to have a recognized quality management system. And that what a quality management system is, for example, ISO 13485 is the standard, the medical standard for developing and manufacturing, commercializing medical products. And what this is what a quality management system is. It's really just a series of procedures that you as an organization will follow to make sure you train your staff, you monitor your calibration of your pipettes and your balances and your equipment, your purchasing specs, your how you do the design and development, and then how you do your record keeping, manufacturing audits, and how you keep design control procedures. For this, I would recommend you get a quality management. If you haven't got a quality management system already, I would recommend you get an advisor to help you implement a quality management system. Of those, the ones that are absolutely essential for all medical products are these 4 or 5 standards. So there has to be a risk management process in your organization. You have, in order to launch a product, you have to follow ISO 62366. So you have to demonstrate usability, and you have to then also comply with the instructions and the information that you give to the customers. So those ones are absolutely essential. That you need as for ISO standards that you need to follow for every product. Plus, you need ISO 13485. All other standards that govern the safety and efficacy of the product vary depending on what the product is, where it's used, how it's used, who on, who by, etc. And we call that the labeling claims. And I'll come to that in a bit as well. So this is where the project. So for all projects, the most important document or the most important thing we believe, or I believe, that you can do when you're starting a project in diagnostics or in medical devices is to create an intended use statement. And if you ever open a packet of ibuprofen or paracetamol or any kind of medical product, there will be an instructions leaflet, and probably on page 1 or 2 there will be an indications for use or an intended use statement that describes what the product is being used for, how it's being used, who are the users, what the patient type is, what material is being measured, where it should be used, and any contraindications or limitations or exemptions. So where you shouldn't use it, for example. So I've done 2 examples here just to show you. So this, because I'm going to walk you through how you go about this. So if you look at, these are two currently commercial glucometers for measuring glucose in blood. And I've highlighted, depending on how it's used, where it's used for, all that, I've color coordinated some of these statements. And you can see, although these are both glucometers, they have very different intended use statements. So the one on the left, for example, in red, it says it's for use in fresh capillary whole blood samples drawn from the fingertip. If you look at the one on the right in red, it says it's for use in venous whole blood, arterial whole blood, heel stick neonatal, or fresh capillary whole blood samples drawn from fingertips. So instantly you can see that the one on the right has got a broader intended use. It can be used in multiple areas. And so then obviously from that, when you're developing this product, you need to provide evidence that that is going to be what you can claim as your labeling claims. And then in addition, it talks about who the intended users are. So if you look at in green, it says the one on the left is for use at home and potentially in clinical settings. Whereas if you look at the one on the right, it is only for use by multiple patient use in professional healthcare settings. And the one on the left is only used by a single patient. So these are very important differences in when you're designing your product. So it's very important to create an intended use statement of how the technology is going to be used and what it's going to be used for, because that will define the level of risk of the product and its classification. It will define all of the developmental activities and clinical trial activities that you need to do to develop this product. So when you're thinking about writing an intended use statement, and this is what I encourage everybody to do as the first thing in all projects, is to basically think about, ideally do some market research or clinical engagement. So talk to clinicians and you potential users, how this would be used, where it would be used, how it would fit into the care pathway, do some market research, and then think about also the launch planning. And that might come out of your market research, but because sometimes you can phase your intended use scope. So you start with an easier intended use so you can get to market, and then later you can expand your intended use with more evidence and extended submissions. But at least it gets you onto the market and starts to get you some money. The broader the intended use statement, the harder it will be to do the regulatory submission, and the more data you're going to need to provide to demonstrate that you are safe and effective in all of those different variable environments. I recommend don't get too excited at this stage. A lot of projects we see, the intended use statement is too broad. It covers all types of cancers, for example, in which case it's not very manageable and it's not very realistic to provide the evidence. Once you've written the intended use statement, you then need to create what they call a design inputs document, and all of this design intent is very important. So for all projects, at the beginning, ideally you want to list all of the design specifications that you are going to meet for this product, and in order for it to satisfy its intent, safe and effective use in the intended use setting. So this might cover areas of robustness, cost. So for example, you might say for robustness, if it's for use at home, then somebody who the user is, are they likely to drop it? How high will they drop it from? How many times do you think they'll realistically use the product for its life, for its shelf life? And all of that sort of activities, what power consumption is needed, whether it's battery or electrical mains, the performance of the assay, what is an acceptable level of sensitivity and specificity or clinical accuracy or diagnostic accuracy, how will it be used, and then also understand the normal use conditions. So for example, you might need it to be stable at different temperature regimes, because if it's being used in a home, there'll be maybe no air conditioning, whereas in a hospital it might be much tighter control on the conditions, the intended use environment. So whatever is important for the adoption, use, safety and efficacy needs to be written as a design inputs, and for each of those design inputs you should list the acceptance criteria. So what is acceptable and what for this project. So stability, it needs to be stable for 12 months at room temperature, or 12 months in a refrigerator at 2 to 8 degrees C. So those are the kind of specifications you will lay out at the beginning of the project. So again, having come up with the intended use statement, you now write the design inputs and the design specifications for this project. This is really important because both of these two documents, the intended use statement and the design inputs, will feature in your technical file reviews. And so the design inputs, as you're going through the project, you will also be following up and writing down the evidence of where you've met those design inputs. So you will be recording under what they call a traceability matrix. You'll be recording the design outputs. The intended use also helps you to write a risk management. And I mentioned to you the 14971, the ISO 14971 regulatory standard for risk assessment and risk management. That is mandated on all products. So you have to demonstrate compliance to 14971 for all products. And that's really about how to manage the risks of your products. So you start with a risk management plan based on the intended use. So you can imagine, as I mentioned to you with those Roche, the two glucometers, sorry, the risks associated with a professional person running the test and a home user measuring themselves have got different levels of risk, because if somebody misdiagnoses, then they at home they may administer a medication which could cause complications, whereas at least if they're in a hospital environment or a professional healthcare environment, then the risk is mitigated because the clinicians will be on hand to deal with the risk. So you're looking at risk and the probability of the risk and the hazard of the risks. And you will be reviewing this throughout the project. So you'll need to keep revisiting the risks of the product. And the idea is to design those risks out during the development process. If you can't design them out because it might be fundamental to the type of product it is, then you need to incorporate that into your instructions, so it's absolutely clear that everybody is aware that there is a significant residual risk of that product. So the risk assessment process will also generate more design specifications. And again, this is critically important. You will need to provide to the FDA or to the notified bodies, you will need to submit the risk management plan and the product risk assessment. And the risk management plan is all about probabilities of occurrence and, like I say, the hazard, the level of hazard. So often it's useful to have clinical input on that to explain what the risks of if this diagnostic test goes wrong. Okay, so up until now, you've set up the project. You've got an intended use statement, you've got your design inputs, you've got your risk assessment, you've understood your regulatory position and what the regulatory standards are, because there are hundreds of regulatory standards, different ISO standards, and they change and like I said to you before, the ingredients differ depending on each project. But basically, you can go on to databases to look at the standards and have a look at what standards are relevant for your type of product and those kind of environments. So if it's a self-testing product or if it's a whole blood versus finger stick, might have different levels of scrutiny and different levels of compliance requirements. I always recommend to start planning the clinical trials early. If you're an academic clinician, then clinical trials is usually a lot more straightforward to oversee. But in terms of a lot of businesses, the clinical trial process takes a long time. It can take a year to set them up. So it's worth starting writing the protocols early. Now, in some cases, for PMAs and for de novo products in the US, you need to agree the clinical trial protocols in advance with the FDA. So it makes sense to engage with FDA as early as possible on those type of projects. I'll come to that in a minute as well. And then at this stage, early in the project, you're doing the test chemistry optimization, electronics design, if there is an electronic instrument or an electronics element to this diagnostic test, software development, and also the algorithm development. So understanding the interpretive software, how the biomarkers or the patient information or whatever is going to be processed to generate the diagnostic statement. So early in the project, the development program, you should be working on these areas. Then once you start to lock down on the chemistry and the software and the hardware, you then pull all those things together. So if it's a lateral flow test, you might have the lateral flow strips made and diagnosed. You might have the plastics, you may have the dropper bottles, all of those kind of elements that you need to bring together. Or if it's a PCR kit, it might be the multiplex different reagents put together and running them as one system. You also start looking at packaging, the packaging that the product's going to go into, the technology is going to go into. You initiate the injection molding tools at this point. So at this point, you might be maybe 8-9 months into the project, and you also, if your product needs calibrators or controls, and I didn't point it out, but on those 2 glucose tests I mentioned before, one of them had calibrators and controls because it was for use in a clinical environment, so they need to register the calibration. The software and hardware for the analyzers will be complete at this point, and then also so that all the software will be fully written and functional. And at this point, you're integrating the whole system of everything. So you've pretty much got design freeze on all of the components of the test, that they're all functioning properly, and that is now a prototype product. So once you've got that prototype product, and this usually can take anything, depending on the tooling and the complexity of the instrumentation, but maybe a year and a half to 2 years. If it's a complicated system, if it's a lateral flow, it might take a year to get to this point. At that point is when you start the manufacturing validation. So one of the common mistakes is that people go to clinical trials before they've done the manufacturing validation. And the problem with that is the final clinical performance and the final verification and validation, i.e. the labeling claims and for the product, have to be demonstrated on the product, not on the technology. And the product basically means that what the customer will buy and will see. So or the end user. So that's why you do the manufacturing validation. So you produce manufactured product that is validated for manufacture, that you have all the specifications, the procedures, the traceability of the manufacture, all of the processes of how to make this need to be put in place before you do your regulatory clinical trial. And it's the same before the final verification and validation. So the data that you generate from the clinical and the V&V work can be used in a regulatory submission and traceable. So the technical file will contain a lot of that information that I've already said, the design inputs and intended use statements, risk assessments, but it will also include the V&V work. And the V&V, the verification of these are main examples of typical of what typically goes into a technical file. Oh, I forgot to mention, at the beginning of the project, you will also write a performance evaluate. Once you've got the intended use statement, you will write a performance evaluation plan where you're listing the studies and the work that needs to be done to demonstrate all of the labeling claims and the risks and the design inputs. But in essence, the main areas that you will focus on for providing evidence, and for this you write, it's good practice and normal practice to write a validation protocol before you do the report, and you sign that off. So you write a protocol to say that we're going to measure stability, or the live testing studies, stability of a product. This is how we're going to measure it. This is what is the acceptance criteria for stability, and it's going to comply with a particular standard for assessing a CLSI standard for measuring stability, for example. So and then you do the validation on manufactured product. So you test products that have been manufactured, usually multiple lots. So you'll test it to show that the product is stable over multiple lots. But you also do the reproducibility and linearity, limits of detection on certain types of technologies, interference studies. Interference studies is very important. There are standard interference studies that you do, things like lipids and cholesterol and paracetamol interference, but there are also product-specific interferences based on the type of patient that you're measuring. So it might be patients who are on chemotherapy, in which case you need to incorporate some of those because they may interfere with the actual test chemistry itself. Looking at operating conditions and the workflow, so studying how the product will actually work, and if people are delay, they get the product out of the freezer. If it sits around for too long, how stable is it? And what if they don't add that reagent in time? What happens? So you need to do a lot of that robustness type work to show, and then at the same time distribution robustness. Again, depending on where this is being sold, if it's being sold to a home user, then you need to demonstrate distribution in mailing direct to users or wholesale to distributors. So there's different levels of robustness that you need to test for distribution testing, but also drop testing and things like that you need to demonstrate that the product will stand up to being dropped. Electrical safety testing is done externally, normally by an electrical safety test house, that they demonstrate that the electronics in your system complies with the appropriate emissions and radiated immunity testing. So they don't get interference from other electronic devices. If your product is sterile, which does occasionally happen in diagnostics, but it's more common in medical devices, if your product is sterile, then you also need to have sterility validation, and that's quite involved. There's standards relating to sterility, and you need to comply. It tells you, and a lot of that's to do with packaging integrity, and also making sure that the sterilization process is adequate. And in certain products, if your product is sterile, then even if it's a low grade of risk for product, you will still need to be audited for the sterilization process to make sure that's been validated properly. So even if it's a self-declaring product, the actual sterilization will probably need to be certified. Usability, and then the software validation. And so all of that will be created to demonstrate what we call the labeling claims. And that's, if you ever look on the inside of an instructions manual, it talks about the levels of performance of the product. It talks about the operating range. It talks about the instructions of how to use things with certain warnings. You generate that information based on all these studies. Then you're going into clinical studies. Now, clinical studies can run in parallel with the V&V. So the clinical studies I have to warn you, they depend on the actual product type and the application, obviously, but typically I'll give you some flavors of the kind of work that you need to do. For clinical chemistry applications where you're measuring a particular biomarker, maybe a known biomarker for, let's just say, PSA, PSA, let's just use that, then you're comparing against a predicate device like a lab reference method. And for that, typically, you only need about 120 patients. So it's not huge clinical studies needed for doing that kind of work. And you're doing a Spearman rank correlation with the predicate device to show that it is as accurate as the lab reference method or whatever the reference method is. With Europe and the UK, it is acceptable to have one clinical trial site for that. For the US, they expect 3 sites in total. But even with those 3 sites, that still can be 120 patients in total. So it might be 3 smaller studies, but it still needs to be done within the USA. They will accept one, normally the FDA will accept 1 European site out of the 3, but you certainly need USA presence in those testing sites. For certain other products, like oncology is a classic one, where your labeling claims are based around sensitivity and specificity, then obviously you're comparing to whatever the gold standard is, whether it's biopsy or MRI or whatever. And those kind of studies have to be, the size of the study has to be powered by the prevalence, and that prevalence is based on where the test is also being used. So if it's based on a community-based screening, then obviously the prevalence is very low. But if this is testing on a biomarker on patients who've already been referred into the hospital, then the prevalence will be higher. So you really need to do the power studies on confidence intervals, etc., to determine the size of the study. But typically, they're about 400 to 500 patients would be the normal range. Again, in the US and UK split also, and Europe, I mean, split is very similar to the one above. And then in terms of other clinical studies, in many other tests, you also need to do normal healthy reference intervals. In fact, this isn't just clinical chemistry, where you will run healthy donors to demonstrate the clinical cutoffs for healthy. It's not, again, this isn't on every project, but it's very common that you will need healthy reference range intervals. So you compare the biomarkers on normal people versus the intended use patients to show that there's a discrimination. In addition to the accuracy studies, which are the ones I've mentioned before, precision is also another main strand of labeling claims. And there are CLSI standards for dictating how these are set up. But basically, typically there's a 20-day study, and that includes multiple lots of cartridge of test, multiple instruments if you have instruments, morning and afternoon replicates, multiple operators, and then you, depending on the kind of product, if there's different clinical cutoffs, you need to do the precision at those clinical cutoffs. You can also use control materials for a lot of these studies. So, but these studies, it's typically 20 consecutive work days. You don't need to work over the weekends to do these. So take about a month to do these studies. And then the data is analysed of the overall imprecision across all of those variables, but also within each of them, to make your labelling claims. So you will often see in instructions for use, it will say precision claims within lot precision, overall imprecision, etc. So that's how you do those. As an alternative, with the FDA, they will accept 3 5-day studies, but these need to be done in the intended use setting and not by the manufacturer. So those studies can be done as part of a bolt-on to the end of your clinical studies, for example, where they could run the 5-day studies. They have both of these options have different merits. 20-day studies are difficult to do. They take a month, but they take a lot of effort, and they can go wrong very easily if you don't concentrate. And then finally, sometimes you will also need, well, not sometimes, you will often need whole blood precision studies. So this is where you're measuring replicates on the same patient to demonstrate the same patient sample, to demonstrate that the product is reproducible. And the size of those projects varies. Okay, so for some of the other validations, I mentioned about de novo, the clinical for de novo and PMA products, then in the eyes of the FDA, they're classed as the highest risk. So even if it was in the FDA's eyes, a class 1 medical device, glucose meter is a really good example. A glucose meter that I mentioned before, I believe, are class 1 medical devices in the eyes of the FDA. Because it's fairly passe and technology and common technology. If you introduce a non-invasive glucose, for example, that is classed as a de novo product. And in the eyes of the FDA, they treat that as if it was a class 3 medical device. So you can see the intended use and the detail of the product can have a massive effect on what level of data you need. So with a normal glucometer, you actually don't need to do a full technical file submission to the FDA. But if you do a non-invasive glucose, you have to do a full de novo. So that's the same as submitting for a pharmaceutical, for example. It's highly controlled. So it's very important to get involved as early as possible with the FDA. if you have your products, and there are websites that you can contact, the FDA website. You can contact them. They have a free service to give you advice of what your classification of your product is, but you need to do that once you've written your intended use statement and you have a clear idea of what the product is going to do. Software validation needs to be done. The actual validation needs to be done on a validation cohort on virgin patient data. So you haven't used that to generate your algorithms. And then depending on the intended use of the product, some of the clinical studies need to incorporate a difference between self-testing, i.e. naive users versus professional users, whether they're trained or naive or untrained. It depends on the type of product. And then obviously you compare that to the lab reference method. So sometimes you do three-way comparisons to show that the users at home are just as good at measuring their blood than a professional user, or the lab reference method on the same patient sample. You can also use biobank samples. This can be done provided it's compatible with the intended use. The FDA don't like this particularly because they like to see work done in the intended use setting. But in some cases, you can use, for CE marking certainly, you can use DNA and RNA extracts from samples, provided they've not been used to generate your algorithms and they are virgin samples, then you could use those to demonstrate your clinical performance. And then likewise, some of those samples could also be run back at the manufacturer's labs. So it doesn't have to be done in a clinical lab itself, but the samples do need to be collected and processed properly by the clinical facility. Okay, so once you now have the verification and validation, and you've got the clinical data, you should also be at this stage preparing your instructions for use and your labeling. My advice here is always start early. This takes a lot longer than you think because there's a lot of editing and change control that needs to be done. But basically, it is a requirement to provide, as I mentioned to you at the beginning, those several standards are required for the information that you provide with your product. So that's on the regulatory symbols. So you'll often see a regulatory label, which is on the bottom right-hand corner here, for example, where it has all of the symbols, the EC representative, the manufacturer's details, and the lot-specific details. That information is essential on the outer box of the product. And then the operator's manuals and everything else, you are allowed to use electronic copies of this, but it needs to be easily available. And then I'll talk about languages in a minute. So the instructions for use need to be prepared in a way that are written so they're easy to use. We would typically encourage you to do out-of-box training, so you allow people to look at the people who've not seen the product before, so intended users, nurses or whatever, to take the instructions, read them, and see if they can run the test out from using the instructions, because that's the aim. And the language that's needed needs to be written sufficiently simple to be able to be understood easily. Languages across the European zone, there are mandated languages. So in order to sell in each of the territories, if you're going to market your product across Europe, you need to provide your instructions in the relevant languages. So if you're selling in Austria, you have to provide German. If you're in Belgium, you have to provide German, French, and Dutch, for example. So that needs to be considered when you're budgeting and when you're planning your rollout plans, that your instructions for use, but also your instruments, if they have a user interface, you need to consider the languages in the user interface. Typically, we do two-stage translations. We do a first medical translation, and then we get the translation itself secondary validated by an independent translator to check that it's been translated correctly. Hard copies, as I mentioned before, are not always essential to provide with the actual product. but the e-copies must be easily retrievable. Now, that's true unless it's things like for home use, you need to be able to provide them with the leaflets in home use. And so just to make it so it's obvious, so it's absolutely certain that they've received the right instructions. Okay, so at this point in the project, you will have probably run between 40 to 50,000 tests to generate the data that's needed to fill a technical file. And so the technical file basically is structured as I've written it here. So there's the device description and the specifications that I mentioned to you before. The labeling, so you actually provide a hard copy of the instruction manual and any instructions and any labels that are provided with the product. You put in all of the design and manufacturing information, so the manufacturing, the overall technical documentation for your manufacturing, all the SOPs, for example. The safety and performance requirements, so you will show a review of the standards and which standards you're compliant to and where the evidence for that compliance is. Risk assessment and risk management plan you'll be submitting. Then all of the the verification, validation, and the clinical study technical reports. So you provide a written report of all of the studies that you've done to show that you're robust to altitude, robust to clean distribution testing or cleaning or whatever. And then the traceability matrix, which I mentioned to you, is about the design inputs and design outputs. You will make sure you provide that, and then you will have the essential requirements checklist. So that's basically all of your outputs against the regulatory standards. And then finally, you'll submit a draft declaration of conformity, which basically is just a statement to say that you meet all of the regulatory standards for this product. Okay, so that's submission ready now. You've got the product submitted. Now, so that's really taking you from end to end at this stage. Really, I just want to spend a few minutes looking at some of the territory considerations in terms of, when you're deciding to commercialize a product. The CE region, the European Union region, requires most products, to have a technical file review through the notified body. You will also need ISO 13485, so I always recommend that you use the same notified body to get your 13485 accreditation as the same, and they're the same. So let's say TUV, they will do your ISO 13485 certification to show you've got all the procedures in place, but they will also then review the technical file of the product. So typically it takes about 12 months to get a technical review slot put in and about 12 months to get ISO 13485 accreditation. So you need to start booking these things early. And then looking at some of the countries, Germany, in my experience, Germany, Italy, and France are very good early adopters, and they have good EU distribution networks. So launching, and then also I've previously had a lot of experience in more of the Eastern European countries as well, who have good distribution networks too. If you're a non-EC European Community company, then you will need an EC representative. Now, in terms of the UK is very slow and difficult, in my experience, to get adopted for diagnostics. So it's quite commercially quite risky. However, it's easy to get into the market because it's self-declaring at the moment. You still assumed that you have all of the data that I've described before, and you still need 13485, but it is easier to get at the moment to get a CA mark than it is to get a CE mark. So, but you will still need a UK responsible person if you are a non-UK company. The UK is also very good, I have to stress, very good for running clinical studies and using the fact that the NHS data is very available. So you can use the good for health economics and that kind of assumptions, because the patient demographic information is freely available online. The US, obviously, you still need to have regulatory approval for most diagnostic products in the US, and that's usually a 510K. But I would recommend you get a a Q sub advice. So that's where you engage with the FDA early. Once you've got your intended use and your performance evaluation plan written, you approach them. The US is half of the world's healthcare budget, and it has a very highly mature distribution network and diagnostic network, and there's a lot of early adopters. And there's also good reimbursement channels. So it's a very clear way of getting into the market. And because distribution is relatively easy to obtain, you can drive quite quick growth. However, it takes about 12 months to gain accreditation from the FDA, provided you have all of the technical file stuff ready. So I would always recommend you use an FDA advisor, maybe an ex FDA assessor. There's quite a lot of them out there who can help navigate you through the regulatory submission. As an alternative, you can launch products under the lab developed test market. So this is using commercial clinical testing labs as a way of using your product to test patients. That still in Europe, that still requires a CE mark under IVDR. And in the US, you can launch without FDA approval if you go through this route. So you can work with companies like Quest or LabCorp or whatever. So you could look into that. However, that in the US is only available for the next few years, most likely, and is probably going to change over the next few years. So you need to keep an eye on that. And then finally, I'm just going to talk a little bit about commercial models. So typically, as I've mentioned, distribution is one of the main areas to launch. You can either go through the main distributors, people like Henry Schein or Cardinal, McKesson, and they will typically take about a 30 to 40% distributor cut. And they will manage the actual placement of the sales and the repeat sales, and usually the basic complaints handling processes from the customer. You could also do a more strategic partnering, so i.e. an OEM in distribution where you sell the product under their name. So this might be under, let's say, Thermo Fisher or Roche. It might be branded under their name, and they're effectively selling your product, in which case they may take between 30 and 50% cut. Often they'll have a secondary distributor themselves, but also that usually includes them doing the marketing and the promotional work for your product. So whilst you're giving away a lot of margin, actually this, for a lot of startup businesses, in my opinion, this is actually one of the best ways to get to market. You can, in the US and in other countries, have what they call manufacturers reps, where you sell direct and you have people selling direct. That's quite a big infrastructure of those manufacturer reps in the US. It is good. You get a lot more profit. You're giving yourself an extra 30 to 40%, and the sales is only based on commission. But it can be risky, slow, and it's quite difficult to manage. Finally, I just mentioned about reagent rental. Often with diagnostics, you have an instrument and a disposable test system where but the instrument might be prohibitively expensive. So what you can do is you can set up a reagent rental where you rent them the equipment and you provide them on a servicing agreement, a fixed number of tests that they need to buy, or on a sliding scale. So you can basically amortize the cost of the capital. So that means that you can take the cost of the instrument out of the equation as a barrier to entry. You're not allowed in the US to give away instruments for free. That's called inducement, and there is an inducement law to stop you from doing that. So you have to give them, if you're going to give them away, you have to do it at cost. You cannot give away below cost. Otherwise, that's inducement. So you can't give them away. But this is a good method of allowing you to predict your revenue, and if you're a startup trying to look at an exit valuation, having reagent rentals in place means you're locked into certain revenue models. And then I've just got in here about the costing. So just to show you how it works. So typically, if you look at the reimbursement for a test, maybe I'm just using this as 40 euros, for example. In the US, the physician themselves will to prescribe this test, they will take a percentage cut, usually 40%. The distributor might take 20%. or 30%, as I mentioned before. OEM might be 30%. And so you can see you need to slice these out. So in order for you to make money, you should be selling the product at 11.76. If the end user price, if the reimbursement is 40, and then from that you need to be able to manufacture your product for, let's say, half of that in order to get a 50% profitability. So this is a good way early on in the project to look at your technology to see whether it's commercially sensible and what the requirements are for your manufactured costs in order to make this a viable product. Obviously, if you've got a new technology there isn't a reimbursement code for, then you have to apply for a reimbursement code, which is very doable, but it can be quite slow and risky. But there are advisors in the US who will help you to apply for reimbursement codes, but you will need to provide detailed costing, and you also need to provide strong evidence of clinical utility. So just as finally, as a wrap up, just typically to give you an idea of roughly how long these projects should take you. So most sort of lateral flow or PCR reagent kits or ELISA kits, for example, should take roughly 21 to 22 months from end to end. Software and the medical devices is usually a bit quicker. And if it's an instrument with a full diagnostic system, that typically should take 3 to 4 years, depending on the size of the instrument. The complexity could take longer. So that sort of gives you an idea of how it all works. So thank you for that, and hope that was useful. And I've got time for any questions.
Thank you, Matt. That was quite a lot of information, and I would like to open now the floor for questions. Are there any questions from the audience? If not, perhaps I would start. Just let's say as a wrap up for someone who is really not so familiar with the process. You mentioned a really good starting point, so the declaration, but are there any helping tools and checklists that could make this process easier and more manageable for really someone who is not as familiar with it as you are?
Right. So, okay. So one of the first things is not really. That's the trouble. So, I mean, there are obviously the IVDR standard, regulatory standard, lists all of the main essence and requirements and the rules. So there's, as I mentioned in the slide in that thing, there's like 22 rules that you need to focus on in the IVD standard in order to submit for an IVD product. So you go through those, and then you need to look at the individual ISO standards. So if you go on to, we use BSI here, but there are databases of the ISO standards, and you can search those by just type in precision or type in clinical studies, and you will get the regulatory guidance for the guidance documents for setting up a precision study, a 20-day study. So one of the EP, I think EP09 CLSI standard. And so you can go to those to look at it. But like I say, you have to start with the intended use statement and then consider each of those. The wording on that is very carefully put together because you need to decide where the product is going to be used and who it's going to be used on, because if it's for home use or if it's professional use only, very, very different levels of evidence that you need to provide at the bench. And it's just different because there isn't really one typical product. So that's why you can't have one definitive list of standards, because every product actually has different standards it's required. The other thing I often recommend is have a look at competitors or similar products. So if you're developing a PCR kit, have a look at a Thermo Fisher PCR kit. Just see what labeling claims they're making and what they often will quote the regulatory standards that they're compliant to. The FDA's website will also list the other people who've submitted 510Ks. So you can look at what submission data was provided to the FDA for that product.
Okay, thank you very much.